Hello, everyone. My name is Yannick Delpu. I'm the Scientific Affairs Manager at Bionano, and I will be your host for today's event featuring Dr. Eric Letouze from the Cordelier Research Center in Paris. I could not wish for a better speaker and a best illustration of what Bionano can offer to the field of cancer research. I would like to remind everyone that this event is interactive. Please ask as many questions as you want during the presentation. And to do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation, and we will also be conducting a survey at the end. But before getting started, I would just like to take a few minutes to introduce you to Bionano's technology. Bionano brings you the true structure of a genome. A cancer genome is probably the most difficult material you can work with, as it is heterogeneous, highly rearranged, and extremely complex. And despite decades of improvements in next generation sequencing, is still not sufficient to give you a clear picture of how these genomes can be rearranged. And the truth is, it is still far from it. And it's pretty easy to understand when you take into account that 2/3 of the human genome is repetitive, making it extremely challenging for algorithms to reassemble the reads in this region. A consequence is that many structure variants will remain invisible to these technologies. And getting reads of several dozen kbs using long-read sequencing is an improvement for sure, but if we go back to the heterogeneity problem, the coverage is the key. And when you want to address highly heterogeneous tumor with a clonal population, what's the point of spending tens of thousands of dollars in sequencing if you just get a fraction of the information you seek? This is how in 2020 that the identification of clinically relevant structure variants such as BCR, ABL translocations are still done by classical cytogenetics, because looking directly at the DNA molecule remains what is the most reliable and cost-effective in a clinical setting. And this is exactly what Bionano does. We are not based on sequencing, but just like you would do with card typing, we directly visualize extremely long DNA molecules. And instead of using a microscope, we developed a super powerful scanner, allowing you to massively image millions of single DNA molecules in parallel in nanochannels. We produce the instruments, the Sapphire. We also have the chips to linearize the DNA. We have kits for DNA extraction and labeling, and our own free analysis tools called Bionano Access and Solve. Let me explain to you how it works in a few slides. As I told you, our technology relies on the direct imaging of ultra-long DNA molecules. That's why we developed the protocol where we lyse the cells in solution, and then we're using a nanobind disk. We tightly bind the DNA to that disk, protecting it from any mechanical stress. And now you simply wash it, elute it, and it is that simple. The protocol is fast and perfectly automatable. Note that FFP samples are just not good enough to be used with Bionano, as the fixative will shred the DNA molecule. But we do this with fresh or frozen material, no problem with the frozen material. Blood, 650 microliters, blood marrow aspirate, same. Cultured cells, fresh or frozen, 1.5 million, and fresh or frozen tissues or biopsies from as low as 10 milligrams. When we say long, we mean it, as our molecule have an average length of about 400 kb and all the way up to megabase. And that's another order of magnitude compared to what long-read sequencing can do. Once we have this megabase-sized DNA, the next step is to label it. Here, an enzyme will recognize a 6 base pair motif and attach a fluorophore directly to the DNA. And the DNA is also counterstained with an intercalating agent. And there's no nicking or probe involved here. It's a simple enzymatic reaction labeling a 6 base pair sequence that appears every 6 kb on average, and the pattern is unique throughout the genome. Then we linearize this DNA. We use electrophoresis to pull the DNA into narrow channels that are engraved on our chips. This is how it looks by electron microscopy. That's what you have on the top, hundreds of thousands of channels in parallel. The stretched DNA is the bottom panel. The blue signal is the backbone stain with the intercalating dye. And the green you see are the labels we attached to the DNA all throughout the genome. And our software tells the distance between the labels, and we can convert that into base pair. And the pattern, once again, is unique throughout the genome, so we know where the molecule belongs, hence the term optical mapping. Now the instrument will cycle the molecules into the channels over and over and image them over and over until you get the massive yields you need to analyze complex human genomes. You can go up to 5 terabase per sample per flow cell, meaning that anywhere between 100x and 1600x coverage for the same price. The only variable being the time you run the instrument. Once we collect these images, the software will perform a de novo assembly of the molecules, and this is probably the most important step. We align every molecule we image based on the label pattern. The de novo aspect is important because when you work from a tumor material, you do not want to force the assembly to a reference that would not make sense here because your DNA is too rearranged. You would miss out the real structure that way. And we build just what we see. And then, once we have these maps, we compare it to the reference. In this case, the reference is in green. We align our map, de novo assemble, to the reference, and we'll look for differences. And in this case, you see a deletion because the space between the label decreases in our sample. And of course, all the knowledge of the reference still comes along, meaning that we know where the genes are because the pattern, once again, is unique throughout the genome and we know where the labels are. So that way we can detect each major type of structural variation starting at 500 base pair and above. We can detect insertion, deletion, very good at repeat expansion. We detect tandem duplication and inversion automatically at 30 kb and above. Translocation and inversion are really hard to detect with sequencing, and we are very, very good at it. And once again, this detection is done genome-wide and automatically. We also develop a specific pipeline for complex heterogeneous genomes that leverages the tremendous coverage and look at structure variants at the single molecule level. And this is crucial when you have clonal population like in leukemia. This tool is complementary to the de novo assembly, and it allows you to detect all structure variants at the level of single molecule, 5 kb and above, automatically, genome-wide, down to 1% allele fraction with a 90% sensitivity. And that's a game changer, and no one else can do this. So now you can visualize your genome in our browser called Bionano Access, and you can see here a circle plot where each chromosome are shown in a concentric circle. Different type of SVs are present as dots. The purple line in the middle are the translocation between each of these chromosomes. If you just click on one of these events, you zoom it to a map where you can see the map aligning to each chromosome. So that's a translocation here. And you can also show the molecules imaged with our system. But on top of that, all these SV calls will be filtered with a control database. So you can just filter only somatic events, rare events that are not present in the control population. And of course, the annotated SV call can be exported as a standard VCF file. Optical mapping has raised a tremendous interest in the cancer field, whether it is to modernize half-a-century-old cytogenetic methods such as karyotyping, if it's to quickly identify actionable SVs such as NTRK fusion partners from tumor DNA, but it also leads to numerous discoveries of new fusions never described before. And let me just show you one last example. I'll finish on an example of a comparative study led by Alexander Ocean at the Radboud Medical Center in the Netherlands. They aimed at comparing the performance of Bionano against classical cytogenetics for the detection of clinically relevant structure variants in a cohort of leukemia patients. The preprint of this work is on bioRxiv, and I really encourage you to check it out. You can also go on our website to have the link to this article. The aim of this work is at term to potentially replace this very complicated cytogenetic workflow you have on the left, requiring a lot of different technologies with a more simple one, more automatable, and less demanding in terms of manpower. And Bionano has the potential to bring them all together in a simple, streamlined assay. The cohort they studied includes both simple and complex leukemia cases, complex meaning that they bear over 5 aberrations. On the right, you have a detail of the average SV count per sample. The take-home message of this study is that this team is the first one to report a systematic comparison between Bionano alone and cytogenetics methods, and they found 100% concordance with the standard of care, meaning here a combination of karyotyping, FISH, and chromosome array. Here is an example of a circles plot for one of the samples. The panel B on the right is a zoom on the balanced translocation between chromosome 11 and 14, and panel C and D are the detail of the assembled map and the molecule detail. This is a case of a complex chromosome 8 chromothripsis structure that was resolved in an AML sample. The circles plot illustrates the shattering of the chromosome 8, called as intrachromosomal translocation here. That's what you see on the left panel, and you have the detail of the map on the right panel. And finally, this study led to the identification of translocations, leading to potential novel gene fusions, which is extremely interesting since these are very important drivers of cancer development. They identified 23 potential gene fusions, of which only 4 were previously observed. This study is still ongoing, and they will include more patients to reach a cohort of 100, and we are very excited to see the final results. But now, I would like to introduce our presenter, Dr. Eric Letouzet, leader of the Computational Center Cancer Genome Analysis Group at the Centre de Recherche des Cordeliers in Paris. Eric and his group develop innovative computational approaches to extract biological insight from large cancer genomic datasets. And his current project involves the analysis of mutational signature and the identification of new driver events in cancer. And today, he will present us the identification and characterization of complex structural rearrangements induced by replication stress in liver cancer. Dr. Letouzet, the floor is yours.
Thank you very much, Yannick. So today I'm going to first show you how we identified a molecular entity of liver cancer with intense replication stress, and then how we used a combination of whole genome sequencing and Bionano optical mapping to unravel complex rearrangements induced by replication stress in these tumors. In the lab, we worked mostly on liver cancer and in particular hepatocellular carcinoma, which is the 3rd leading cause of cancer death in the world. And it has a very diverse incidence in the world related to the diversity of risk factors. So the main regions for HCC are Southeast Asia and Sub-Saharan Africa, due to the prevalence of HBV infection together with exposure to the aflatoxin B1 toxin. There is also a high incidence in Japan and Egypt due to hepatitis C virus infection. In France, the major cause of HCC is alcohol consumption, and in the United States, the dominant cause is non-alcoholic steatohepatitis and other metabolic disorders. These risk factors induce HCC mostly because they will damage the liver, leading to fibrosis and later a cirrhosis of the liver, and cirrhotic nodules can evolve to dysplastic nodules that will progressively transform to HCC and advanced hepatocellular carcinoma. But there are other cases in less than 20% of patients that develop on a non-cirrhotic liver. And one of the mechanisms for that is the insertional mutagenesis due to different viruses. Of course, the most important virus is HBV. It has been shown that HBV can insert into the human genome, and when it inserts near an oncogene, it can lead to the overexpression of the oncogene and cancer development. And in the lab, we have identified another virus, AAV2, that rarely can integrate in the genome and also lead to the overexpression of oncogene and HCC development. And recently, we identified a new molecular entity of HCC that develops in non-cirrhotic patients and is driven by the activation of two different cyclin genes, namely Cyclin A2 and Cyclin E1. We discovered this subgroup by the analysis of 750 HCC with a combination of RNA sequencing plus whole exome or whole genome sequencing. With this approach, we identified recurrent events leading to the activation of Cyclin A2 that are summarized on this graph. These events can be viral insertions by either AAV2 virus in orange or HBV virus in green. Here, each arrow represents a viral insertion identified in one patient. But we also identified some recurrent gene fusions that are depicted above that also involve the same INSHN 2 of Cyclin A2. And the functional consequences of all these events will be to generate a truncated Cyclin A2 protein starting at the beginning of exon 3 at methionine 148 or 158. And this protein will be constitutively active because it will lack the regulatory domains, destruction box and ubiquitin domains that are on the N-terminal part, while keeping the functional cyclin force on the C-terminal part. So these are new mechanisms leading to the activation of Cyclin A2 in HCC. We also identified recurrent events leading to the activation of Cyclin E1. Again, it can be viral insertions by AAV2 or HBV virus, or structural rearrangements up to 50 kb upstream the transcription start site. And we verified with RNA sequencing data that all these events, both in our cohort and in the TCGA cohort, lead to a massive overexpression of Cyclin E1 with a fold change greater than 100x. These two genes, when activated, will define a homogeneous HCC subclass, as you can see on those T.I.C plots. So these are classifications of tumors in our cohort, LICA-FR, and the TCGA cohort. And you can see that tumors with an activation of Cyclin A2 or E1 are very close at the transcriptomic level and define a homogeneous subgroup. This subgroup has also clinical particularities. It is a very aggressive subgroup with very dismal prognosis. And the CCN-HCC tumors are also particular in that they tend to occur in younger patients with a non-fibrotic liver, and they will be very big tumors, usually greater than 10 centimeters. At the molecular level, this subgroup has an enrichment of inactivation of RB1 and PTEN tumor suppressors, and it is exclusive with the activating CCN-NB1 mutations that are frequent in other HCC. Now, if you look at the gene expression features, this tumor subgroup has a strong overexpression of E2F targets that reflects the very fast cell cycle in these cells. And also, it shows a strong overexpression of ATR pathway that is implicated in response to replication stress. This can be expected because Cyclin E and Cyclin A are both implicated in cell cycle progression, and more precisely, they will cyclin E promotes S phase entry and cyclin A progression into S phase. And it has been well documented in several models that when you activate cyclins A or E, cells will enter S phase prematurely, which leads to replication stress. And we wondered if in this subgroup of tumors, the replication stress that we can detect at the transcriptomic level would lead to a particular type of genomic instability. To assess that, we analyzed the whole genome of 15 CCN-HCC, and we examined first the number of different types of somatic alterations. Here you can see from left to right the number of point mutations, small insertions and deletions, and structural rearrangements in CCN-HCC versus other HCC. So in terms of point mutations and indels, CCN-HCC don't have any particular increase in terms of the number of mutations. By contrast, they all show a very high number of structural rearrangements with, on average, more than 400 events per tumor. So we wondered if this massive load of structural rearrangements could have a specific signature that could be a signature of replication stress. And to test this hypothesis, we looked into more details into the types of structural rearrangements that we could find in these tumors. When we talk about structural rearrangements in cancer cells, we can talk about simple rearrangements with the well-known categories of deletion, duplication, inversion, and translocations that can be reciprocal or not. And as mentioned by Yannick, there are also more complex events that will generate a large number of abnormal junctions in a single catastrophic event. And the most well-known example is chromotripsis, where a chromosome or a fragment of chromosome is broken into pieces into a catastrophic event, and these pieces are then stitched together but in a random order, leading to the accumulation of numerous copy number and abnormal junctions in a single event. What about HCC? By whole genome sequencing, we found that on average, an HCC displays 50 somatic structural rearrangements, but the profiles are very heterogeneous, as you can see with those three examples. So here you can see circus plots with different types of events represented with a color code. The first tumor on the left, CHC 892, is very quiet with just one interchromosomal translocation and a handful of deletions. By contrast, the tumor in the middle has a very large burden of tandem duplications in green and interchromosomal translocations in gray, whereas the tumor on the right has a deleter phenotype with a large number of deletions. So this suggests that there can be different mechanisms generating different types of structural rearrangements that are more or less active in different HCC. And to analyze this further, we used a method to detect signatures of structural rearrangements that is an adaptation of the well-known method for mutational signature analysis. The idea is to split the different rearrangements into categories. So here we divided rearrangements by the type of events, deletions, duplications, inversions, translocations. Also by the size of the events, ranging from less than 1 kilobase to more than 10 megabase. And we distinguished clustered from non-clustered rearrangements. With this approach, we could identify 6 distinct rearrangement signatures operative in HCC. Signatures RS1 and RS2, for example, are duplicative phenotypes with a prevalence of duplications, but that differ by the size of the events. And also the RS1 signature comes with frequent interchromosomal translocations. Signature RS3 is a signature of clustered rearrangements that actually correspond to chromatripsis. And then signatures RS5 and RS6 correspond to deleter phenotypes with different size ranges. Next, we classified our tumors based on the number of rearrangements attributed to each signature, as you can see on this clustering. And this allowed us to identify a group of tumors that you can see on the left that has a very high number of structural rearrangements, several hundreds usually, that are attributed to signature RS1 in dark green. And it turned out that this this group of tumors correspond precisely to the CCN-HCC subgroup. And this led us to conclude that the RS1 signature may be the signature of rearrangements induced by replication stress in CCN-HCC. So let's have a closer look now at this signature. It is characterized by the accumulation of focal tandem duplications, smaller than 100 kilobase, and it comes together with many interchromosomal translocations. It is very easy to recognize in a circus plot with these numerous duplications in green and interchromosomal translocations. And it also leads to a very characteristic copy number profile with a high number of focal gains that are distributed throughout the genome. Now we will zoom into the chromosome 14 of this tumor to have a closer look at these events. Here we are. Here you can see the different focal gains. And on the top of this log ratio profile, we overlaid the structural variants identified by whole genome sequencing. So you can see that some of these gains correspond to classical tandem duplication, where a segment, here segment B, is duplicated just next to itself. This is an expected structural rearrangement leading to a focal gain, but we also have more unexpected mechanisms where a focal gain is actually surrounded by 2 translocation breakpoints, in this case here, going to chromosome 2. And if you go on chromosome 2, you will see another focal gain bordered by translocation breakpoints going back to chromosome 14. So we have here a complex event where 2 focal gains on 2 different chromosomes are interconnected by translocation breakpoints, a mechanism that we called interchromosomal templated insertion. So what could be the resulting molecule from such an event? By whole genome sequencing, you can only reconstruct 2 possibilities. One possibility is that the segment E from chromosome 2 is inserted in the middle of 2 copies of segment B inside chromosome 14. And the second possibility, on the contrary, is that segment B from chromosome 14 is inserted in the middle of 2 copies of segment E inside chromosome 2. With whole genome sequencing, you cannot distinguish these 2 possibilities because they both respect the abnormal junctions that have been detected as well as the copy number for each segment. So if you want to confirm the existence of such an event and to identify the real molecule in the tumor, you need some long read analysis, and that's why we performed an analysis with Bionano optical imaging. So we analyzed in this project 4 HCC and matched normal liver samples. The starting material was frozen tissue with 30 milligrams. And this tissue went through the protocol already explained by Yannick, with an isolation of high molecular weight DNA, followed by labeling with DLE1 enzyme, transfer into Saphyr instrument, and then the barcode of the labels is read by the Saphyr instrument, and the molecules are then analyzed with Bionano software to map them on the human genome and identify structural rearrangements. First, a few technical metrics. We did obtain very high molecular weight DNA from all our samples except one normal sample for which we could not provide enough starting material. We had for every sample around 1,300 gigabase pairs of molecule greater than 150 kb, and with an average molecule size around 250 kb. So we did obtain very long molecules with this protocol, and we were quite impressed by this because usually with our extraction protocol, the DNA is fragmented in 10 kilobase segments. On top of that, we obtained a very good coverage with 300x through the genome, which is a great coverage to identify subclonal rearrangements in tumors. Okay, so in terms of analysis, the molecules obtained by the Saphyr instruments were analyzed by the two pipelines described by Yannick, the de novo assembly pipeline and the rare variant analysis pipeline. And the particularity of our project is that we are analyzing tumors and we are interested in somatic structural rearrangements. So to obtain these lists of somatic structural rearrangements, we compared SVs called in our tumor samples with the matched normal liver samples to identify SVs that are specific to tumor cells. Here are the results. You can see here the circus plots showing the rearrangements identified by Bionano optical mapping in our 4 tumors. We had selected 2 tumors belonging to the CCN-HCC subgroup with replication stress, and 2 tumors belonging to other HCC subgroups. And you can see here that with Bionano, we successfully recovered the RS1 signature of replication stress, characterized by numerous interchromosomal translocations and duplications, specifically in the 2 CCN-HCC. I just want to highlight that in tumors CHC 3798, the second tumor from the left, the tandem duplications were very small, so they were identified as insertions in purple in most of the cases. So overall, Bionano optical mapping successfully detected our signature of replication stress in the 2 samples where they were expected. Because we also had whole genome sequencing for 3 of the tumors, we performed a comparison of structural variants followed by short-read whole genome and Bionano. I must mention that the whole genome sequencing we performed was quite a high depth because we had 90x for the tumor and 30x for the normal. This is Illumina sequencing, 100 base pairs, paired end, and we called structural variants with MANTA software. Here you have the comparison of SV calls with whole genome sequencing versus Bionano in the 3 tumors. The first tumor is classical HCC with relatively few rearrangements, and the 2 others are CCN-HCC with a high number of rearrangements induced by replication stress. In the first tumor, we had relatively few rearrangements with whole genome, and we called a little bit more with Bionano. In the second tumors, we could already see the signature of replication stress in whole genome with a large number of translocations and duplications. but we found even more events with Bionano. For example, we had 78 tandem duplications in whole genome sequencing versus 127 with Bionano. And Bionano also calls other types of rearrangements like the insertions and intrachromosomal translocations that do not correspond precisely to deletions or inversions. In the third tumor, 3798, there was a large number of duplications identified in whole genome sequences and less in Bionano. But this is due to the fact that these duplications are very small and sometimes too small to contain enough markers to be identified as duplications with Bionano, and they are called instead as insertions. So they are identified, but the number of labels is not sufficient to classify them as duplications. Overall, the Venn diagrams show that the two approaches give very consistent results with a large number of overlapping events, but Bionano calls extra events and with on average 1.5 times more events identified than with whole genome sequencing. So the events that were identified by whole genome sequencing and not Bionano are often very focal events, sometimes smaller than 1 kb, and these events are difficult to identify with optical mapping. And by contrast, I will show you in the next few slides what are the numerous additional events we were able to detect. On this graph, you have a zoom on one chromosome with the log ratio profile obtained by whole genome sequencing. Each point is a 1 kb window. And on top of these log ratio profiles, we overlaid the structural variants identified by whole genome sequencing on the top and by Bionano at the bottom. First, you can see they are highly consistent, and then we can have a look at some events that are detected only with Bionano. Some of these events are centromeric, and these regions are known to be repetitive and very hard to analyze with sequencing approaches. Bionano, in turn, is able to identify some rearrangements in these areas. We also identified many subclonal events with Bionano that were missed with whole genome sequencing, simply because the coverage with Bionano is very good with 300x versus only 90x for whole genome. And finally, we identified more complex rearrangements that were missed with whole genome sequencing. Here, the red arrows show you some intrachromosomal rearrangements that associate together different regions on the same chromosome, and this very complex rearrangement was missed by whole genome sequencing. This is exactly the type of events we wanted to characterize in detail, and because we had identified some complex events related to replication stress that we wanted to elucidate. And I will now show you a few typical events related to replication stress that we could characterize in depth. This type of event, I already showed you an example a few slides ago, is called interchromosomal templated insertion cycle. Here you have two focal gains on two different chromosomes that are associated with interchromosomal translocation breakpoints. With whole genome sequencing, we can only imagine the resulting molecule, and there are two equivalent possibilities, one in which segment B is inserted in the middle of two copies of segment A, and the reverse option where segment A is inserted in the middle of two copies of segment B. And with Bionano, we could resolve these two possibilities. Here you can see the different molecules, the long molecules that were analyzed by the Sapphire instrument. And so they are in orange, and the blue dots on these molecules correspond to the marker of DLE1 enzyme. From these molecules, Bionano could reconstruct a consensus sequence, the MAP4381, that corresponds to the local region. And with this sequence, we can reconstruct our complex rearrangements. Here, let's start with the beginning of the molecule. The barcode of labels fits nicely with the beginning of our event on chromosome 3. Later on the molecule, the 4 labels correspond to the B segment on chromosome 3. And you can see that these labels are found at 2 different places in the molecule. So we can see the duplication of this region. And in the middle of these 2 copies, you have another region that this time aligns on chromosome 1 and corresponds to our segment A. And the end of the molecule is just the following of chromosome 3. So with this approach, we can first confirm the existence of this interchromosomal templated insertion event. And we can ascertain that the real molecule resulting from this event is an insertion of a segment A into chromosome 3. With optical mapping, we could identify other classes of recurrent events due to replication stress. And in particular, an important class are intrachromosomal templated insertion cycle. This is quite similar to the previous events, unless that the 2 focal gains are located on a single chromosome, here chromosome 10. You can see 2 segments, one around 79 megabase, another one around 91 megabase, that are both gained and that are associated with inversion breakpoints detected in whole genome sequencing. With Bionano, we could resolve the structure of this complex event and see that segment B is actually duplicated with an insertion of segment A in the middle. So we can reconstruct the molecule resulting from this rearrangement where the A segment has been inserted within 2 copies of segment B at position 91 megabase on chromosome 10. And finally, these are events that involve only 2 segments, but we also have some more complex patterns involving up to 10 different segments that can be located on a single or different chromosomes. And I will finish with this example here that involves 4 different chromosome segments that have been gained in a single event related to replication stress. The first 3 events, A, B, and C, are located on chromosome 3, and the last one, segment D, is located on chromosome 6. And you can see that different abnormal junctions corresponding to inversions or translocations join these different segments. With Bionano, we were able to identify some molecules spanning the complete event and showing that we have an adjacency of segment C, then A, then D, and then B. And so we can reconstruct precisely the complex molecule that arises from this rearrangement with in the middle of the 2 B segments in chromosome 3, you have a juxtaposition of segments C, A, and D. So this is quite exciting because with the combination of whole genome and optical mapping, we can reconstruct very complex karyotypes at base pair resolutions. But the events I showed you are around between 50 and 100 kb. And we sometimes have some events that involve segments smaller than that, in which case it can be difficult to recognize the region of the genome corresponding to each segment because you will only have very few markers. So we are currently developing a computational method to validate some complex events involving focal segments. You have an example here. So let's say you have 4 very small segments that are joined together and each one just has one or two DLE1 motifs, then it won't be possible to map them efficiently with optical mapping. So what we will do is we will reconstruct the candidate molecules based on whole genome sequencing data. Then, since we know the DLE1 motif distribution on the genome, we can reconstruct the expected barcode for each molecule. And we will look directly for these barcodes into the long molecules in optical mapping data to find the reconstruction that is actually present in the tumor. So far, we have been able to characterize diverse types of structural rearrangements related to replication stress with whole genome sequencing and optical mapping. About half of the events in CCN-HCC are classical tandem duplications, and about 40% of events are templated insertion cycles that can be intrachromosomal or interchromosomal, like I just showed you. Interestingly, all these events are consistent with a break-induced replication mechanism, where the replication fork gets stalled at some point and will jump to another location, either on the same or different chromosome, replicate a stretch of DNA, then move back to another region, and finally come back to the original template, leading to these different types of complex structural rearrangements. Finally, we investigated the location of these breakpoints on the genome to find what features could favor the blockade of replication fork. And on this graph here, you have on the first line the density of breakpoints due to replication stress per 500 kb windows. And you can see that the hotspots of replication stress breakpoints are associated with regions that tend to replicate early, to have an active gene expression and active chromatin marks, in particular H3K27 acetylation, which is the mark of enhancer regions. In fact, the genome is divided into topologically associated domains that are active or inactive, and we have a strong bias for replication breakpoints to occur in early replicated active topologically associated domains. This is a graphical summary. With this study, we are able to define a new entity of HCC representing 7% of the tumors that are initiated by Cyclin A2 or E1 activation, leading to premature S-rate entry, replication stress, and replication fork collapse followed by break-induced replication will give this very characteristic rearrangement signature that we observed. And there can be second hits due to these rearrangements, and we identified recurrent third activating events. And this will give large tumors highly proliferative with a poor prognosis. But we hope that this subgroup could be targeted because the replication stress may confer vulnerability, for example, to PARP inhibitors or ATR pathway inhibitors. In conclusion, the optical mapping technology allowed us to identify more structural rearrangements than whole genome sequencing, to unravel complex events induced by replication stress, and may be used to identify replication stress in clinical samples. We found that replication stress in liver tumors generates a specific signature of tandem duplications and templated insertion cycles, and defines a new entity of HCC with specific clinical and molecular features that may be used for targeted therapy. This work was mostly performed by a PhD student, Quentin Bayard, and the tools we developed for rearrangement signature analysis are available in GitHub in our package called palimpsest. And to finish, I will thank all my collaborators in Jessica Zickman-Ross's lab, our collaborators abroad and sponsors, and thank you for your attention.
Thanks a lot. Thanks a lot, Dr. Letouze, for this presentation. We will now start the live Q&A portion of the webinar. And if you have a question you would like to ask, please do so now. You just click the Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. So let's get started. The first question, are all the HCC managed the same way, whether they are originating from a cirrhotic liver or from a viral infection? I mean, in a clinical way, are they managed the same?
So the management of HCC depends on the BCLC stage that takes into account the number, the size of the tumors, and the involvement of and the insertion into blood vessels. For early stages, small tumors can be offered curative treatments by surgery or chemotherapy or radioembolization. But for advanced stages, the main treatments are a combination of tyrosine kinase inhibitors, and immunotherapy are being more and more used. And regarding cirrhosis, the problem with cirrhosis is that it will increase the toxicity of different drugs. So the patients who have a healthy liver, or at least non-cirrhotic, can potentially benefit from more treatment options than the others, and it's the case for this tumor subgroup that we identified here.
Okay. Second question: can Bionano also predict viral insertion in the chromosome? Do you want to answer that, and I can probably
yes.
a bit later.
So the 4 tumors we analyzed had some HBV insertions that we had already identified with whole genome sequencing. The problem is that the HBV virus is quite small, and it doesn't contain any of the GLE1 marks. So the insertions of the virus are detected because they will shift the labels of surrounding human genomic regions. And so you can detect an insertion, but if you don't know if there is HBV, you cannot prove only with optical mapping that this is an HBV insertion.
Thanks. I will just add a little bit to that. There is a non-commercial option to do this using a CRISPR assay that will bind to the DNA and light up the sequence that do not contain DLE marks. So that way you can light up the telomeres, so you can light up viral sequences for which you design guide RNA for. Can you remind what tool did you use for detecting destructive variants from your whole genome sequencing data?
For whole genome sequencing, we used the MANTA tool that takes as input the BAM files from the tumor and normal and identifies the somatic structural variants. We are quite happy with this tool because the structural variants we identify usually map very well the copy number profiles, which gives a positive control for the quality of variant calling.
Okay. Will this approach detect any heterogeneity in the tumor population? I think this question refers to whole genome sequencing on one hand, Bionano on the other, and the combination of the two.
So with whole genome sequencing, we can estimate the proportion of tumor cells that contain each rearrangement simply by counting the proportion of molecules at some genomic region that contain the rearrangement versus the total number of sequenced reads. This gives us an estimation of the proportion of tumor cells bearing each rearrangement. We haven't tried yet with Bionano data to do this, but since we also have a count of the molecules corresponding to both the rearranged events and the normal DNA sequence, I guess that we could just as well compute variant allele fraction and get an estimate of the clonality of each rearrangement.
Just to add up to that, you used a 300x coverage for this study. I was referring to up to a 1600x coverage. This new 5 terabase pair yield that you can get per sample is the latest iteration of our technology that is available since just a few days, explaining how you used 300x coverage for that. Is there a way to reconstruct the complex rearrangements, or has it to be done by visual inspection? I think you answered this in your last slide.
Yes, so for complex rearrangements, I think in Bionano software you can detect when you have a consensus molecule to where regions of the genome aligns each part. But yes, we also did a lot of visual inspection because it was our first project with this technology. And like I mentioned, I think something we added is that when you have
I think we lost Dr. Latouze for a few minutes. Just for the time he reconnects, let me maybe try to find a few questions that are referring to Bionano. So we had a question on does Bionano detect also ecDNA? There was a recent paper.
Sorry, I got disconnected
recently.
I just came back.
Okay. Just finishing the question I was answering on the ecDNA. We do not detect the circular structure of the DNA by itself, and also because the DNA will break during the randomly break during the DNA extraction, but we will detect all the rearrangements that are typical for this type of structure, namely the large amplifications that are associated with this type of ecDNA. I direct you to our website where we refer to this paper, very interesting paper on ecDNA published recently. To go back to the question with Dr. Latouze, a tumor with a cyclin activation presents a high number of structure variants, I believe. So are they acquired in a single catastrophic event, or does this happen progressively during tumorigenesis?
Sorry, I missed the question, sorry.
The tumor with a cyclin activation presents a high number of rearrangements. Are they acquired following a dramatic catastrophic event, or all the way during carcinogenesis?
Okay. So to answer this question, we were able to analyze for a few cases, both a primary tumor and a relapse a few years after. And in each case, we could monitor the evolution of the phenotype with the acquisition of additional events with the same signature. And we also have, in some cases, very complex rearrangements that we interpret as a templated insertion cycle reusing the same segments as a previous one. So I think that the rearrangements induced by replication stress are accumulated progressively during tumorigenesis.
Okay. Would you say they fuel the tumorigenesis, or the accumulation of events is more passenger events leaving genomic scars?
Okay, so indeed, you can wonder if it's the rearrangement signature we see is just a consequence of replication stress, or if it plays a driver role in tumorigenesis. At least we identified one recurrent event in third promoter that is significantly enriched in this tumor subgroup. And for these events, they definitely fuel tumorigenesis because TERT is a well-known oncogene in HCC. But we haven't identified other regions that were altered significantly more than expected by chance in this subgroup. So for now, only the TERT rearrangements can be defined as drivers, but we only analyzed so far 15 HCC and HCC. So I think we need to increase the series, and we might identify additional secondary drivers activated by replication stress-induced rearrangements.
Okay. What would you say is the advantage of combining whole genome sequencing with Bionano versus each methods individually?
Well, I think the two methods are very complementary. The strength of whole genome sequencing is that you can detect the abnormal junctions at base pair resolution. But the weakness is that you only see, you cannot detect complex events in their entirety. Whereas with Bionano, you do not have a base pair resolution, but you can really reconstruct complex events very well. So when you combine both, you can have both the large view of a long molecule as well as the precision of whole genome sequencing at base pair level.
Okay. I think we have time for one last question. I really like the translational aspect of your work, more of a comment, but when you say that Bionano identifies the replication stress signature in your samples, I can help drawing a parallel with the need to identify patients eligible to PARP inhibitors that present an accumulation of DNA damages in breast, ovarian, prostate cancer. To that regard, you mentioned that Cyclin-activated HCC may benefit from a tailored therapy. What are your plans to move forward in this direction?
We have two projects going on to try to move to the clinic. The first is to try to identify CCN-HCC in retrospective cohorts, and to do this, we are testing immunohistochemistry of Cyclin A2 and E1 to be able to detect this subgroup in paraffin-embedded tissue. And the second one will be to develop a mouse model. So we are collaborating with a team at Mount Sinai to develop a mouse model of CCN-HCC that we could use to test the drugs that seem logical for this subgroup, in particular the ATR inhibitors that will directly target the response to replication stress.
Okay, nice. We still have 32 unanswered questions that we will follow up by email because we're running short of time. Thank you again, Dr. Letouze, for your time. Do you have any final comments for our audience?
Well, thank you very much for the opportunity to give this talk. I must say, I think the structural rearrangements will really gain a lot of we will gain a lot of insights from technicals such as optical mapping that is very complementary with the whole genome sequencing. So I'm quite excited about this approach.
Thanks again. Thank you again. And before we go, so thank you all of you for joining us today and for your question. Once again, the question we didn't have time to cover today, we will be submitting by email, and we will get back to you. Once again, I would like to thank Dr. Letouze for his time today and his research and amazing results. Thank you again to LabRoots for making this webcast possible today. This will be open to view on demand, so LabRoots will alert you with an email when it's available for replay. And we encourage you to share this by email with your colleagues or people that have missed that event. A survey will pop up right after we finish, and please answer to help us improve the webinar program. And until next time, thank you and goodbye.